What is time-reversed biology anyway? Imagine watching a movie of a plant growing. You see leaves unfurl, flowers bloom, insects pollinate. Now flip the film backward. Petals retract, sap flows uphill, broken bonds reassemble. Time-reversed biology asks whether life could actually work this way, with organisms that consume disorder and spit out order, effectively unaging and rebuilding themselves by drawing energy from a decreasing entropy environment. Why does life march forward with entropy? Every metabolic reaction in our cells, from digesting glucose to copying DNA, increases entropy in the surroundings, even as it builds complex molecules inside the cell. The second law of thermodynamics guarantees that, on balance, disorder rises. Life locally creates order, cells, organs, ecosystems, only by exporting even more disorder, heat, waste to its environment, making the arrow of time and the arrow of life inseparable under normal conditions. Could a life form actually harvest negative entropy like Maxwell's demon? In thought experiments, Maxwell's demon sorts fast and slow molecules to create a temperature difference, seemingly reversing entropy, but in practice, the demon itself must expend energy and increase entropy elsewhere. A hypothetical time-reversed organism would need an environment where entropy flows backward, a perfect, improbable heat sink that runs ever cooler without energy input. No natural setting offers that, so even the cleverest cellular demon gets trapped by the same entropy tax. What if the universe itself contracts or recurs? Yet some cosmological models envision a big crunch or Poincaré recurrence where the universe retraces its states. In such a scenario, every process, even life, might play out in reverse during contraction. Yet any real organism would need memory and apparatus tuned to reverse microscopic events perfectly, a feat far beyond biochemical plausibility. Instead, we end up with speculative Boltzmann brains, fleeting random assemblages that briefly mirror our thoughts in a sea of statistical fluctuation. Can reversible computing or quantum biology tip the scales? Reversible computing uses gates that, in principle, avoid energy dissipation by ensuring every operation can be inverted. If cells ran on fully reversible molecular machinery, they could minimize but not eliminate the entropy they produce. Quantum systems can flirt with lower entropy fluctuations, yet decoherence and measurement still force entropy increases, so even quantum life, carefully engineered, bows to the unstoppable tide of disorder. What's the takeaway for curious explorers? Time-revered biology throws into sharp relief why life thrives on energy gradients and why the arrow of time shapes every heartbeat, leaf sprout, and memory. While physics permits momentary, tiny entropy dips, a truly backward-living organism remains locked in the realm of imagination, capturing our curiosity, but reminding us that in the real world, we grow, age, and evolve forward, powered by the ceaseless rise of entropy. Time-reversed biology imagines organisms whose internal processes run opposite to the familiar thermodynamic arrow, locally decreasing entropy instead of increasing it. In such life forms, metabolic pathways would rebuild complex biomolecules from waste products, effectively putting broken eggs back together against the natural tide of disorder. This radical notion challenges the assumed universality of the second law of thermodynamics in open, far from equilibrium systems like living cells, suggesting that under exotic conditions, biology could harness negative entropy flux to drive growth and reproduction backward in time. A hypothetical cell, for instance, might absorb scattered proteins from its surroundings and spontaneously reassemble them into functional ribosomes, reversing degradation like ash flowing uphill into a pristine log, as if the embers remembered their original shape. Conventional biology relies on consuming low-entropy nutrients and expelling high-entropy waste, thereby increasing the universe's overall disorder even as local cellular order grows. Time-reversed biology would invert this balance. Cells would import disorder from the environment and export restored order, locally reducing entropy while building internal complexity. Such a reversal would necessitate a surrounding medium with even lower entropy to preserve the global validity of the second law. Humans, for example, oxidize glucose into carbon dioxide and water to fuel metabolism, whereas a time-reversed organism would absorb CO2 and H2O from the environment to reconstruct glucose, akin to unmixing paint by swallowing blended hues and expelling the pure pigment separately. At the microscopic scale, physical laws such as Newton's equations and quantum dynamics are time-symmetric, allowing processes to run forward or backward in theory. Yet statistical mechanics overwhelmingly favors entropy in increasing trajectories, making reversed evolutions astronomically improbable in macroscopic systems. Time-reversed biology would have to emerge from or exploit rare fluctuations where disorder spontaneously self-organized into order, a phenomenon so unlikely that it is essentially forbidden in large ensembles. Reversing random diffusion, for instance, would require Brownian particles to spontaneously cluster into a tight configuration, like shuffled cards mysteriously restoring themselves to perfect sequence. Reverse metabolism, the engine of such life, would involve hypothetical enzymes capable of catalyzing endergonic, order-creating reactions without conventional energy inputs. Instead of expending ATP to link amino acids into proteins, reverse catalysts would harvest ambient heat or random motion to drive molecular assembly, effectively operating as information-powered Maxwell's demons. Realizing such catalysts would require molecules with negative activation barriers, fictional underknown chemistry, and coupling to exotic fields or coherence phenomena that can leverage thermal noise without violating conservation laws. A reverse ribosome, for example, might capture free amino acids from a chaotic solution and spontaneously organize them into complex proteins by siphoning energy from environmental randomness. Much like a shredder reassembling scattered scraps of paper into coherent documents using nothing but room heat. For such processes to be viable, time-reversed organisms would need hypercephalid feedback control systems capable of detecting local entropy increases and triggering corrective actions instantaneously. This demands information engines operating with minimal thermodynamic cost, nearly ideal Maxwellian demons able to measure molecular states without producing corresponding entropy elsewhere. The life forms would have to monitor and reverse every misfolding protein or chemical imbalance in real time, using advanced informational networks far beyond current biological capacities. It would be like librarian who instantly senses any misplacement of a book and teleports it back onto the correct shelf by reading microtraces left in the air. Backward entropy flow systems would exhibit strong nonlinear thresholds below a critical scale, such as in nanoscopic vesicles or primitive protocells. Thermal fluctuations could transiently reverse local entropy, leading to brief windows where reverse metabolism becomes possible. But above these thresholds, ordinary entropy production reasserts dominance, preventing sustained backward life. These thresholds resemble phase transitions in statistical systems, where tiny changes in energy or density can shift a system between ordered and disordered states. A nanoscopic lipid vesicle, for instance, might briefly self-assemble from dispersed molecules under rare fluctuation conditions before dissolving back into randomness, like a water droplet freezing spontaneously in a supercooled vapor under perfect conditions. If backward entropy biology were possible, it would open new possibilities for the origin of life theories, especially in exotic environments such as quantum vacuum fluctuations, supercooled cosmic regions, or near black hole event horizons. In such niches, reversed flux chemistry could seed molecular complexity, enhancing the pathways to abiogenesis. Dual square of time models could explain how life's chiral asymmetries, such as homochiral amino acids, emerged from early competition between forward and backward fluxes. It would be akin to primordial soups that occasionally unboil their own ingredients, producing increasingly elaborate molecular recipes without external chefs. Yet the practical challenges in realizing time-reverse biology are immense. Demonstrating sustained negative entropy flow in any biological or chemical system is effectively impossible with current technology due to the overwhelming odds against the necessary fluctuation pathways. Engineering reverse metabolic enzymes would collide with fundamental kinetic and thermodynamic constraints, and detecting fleeting negative entropy currents amid the overwhelming background of forward entropy production would demand sensitivities surpassing known physical limits. No laboratory to date has measured a stable negative entropy flux in any macroscopic reaction network, making the endeavor akin to trying to catch a shadow unmaking itself in a field bathed with light. Nonetheless, exploring the theoretical boundaries of entropy reversal illuminates important aspects of non-equilibrium statistical mechanics, quantum control, and synthetic biology. Advances in manipulating quantum coherent systems, designing fluctuation-powered nanomachines, and probing the limits of feedback control may one day reveal just how close living systems can approach entropy reversal threshold. Even if true backward-running life remains an implausible fantasy, investigating it sharpens our understanding of why biological systems thrive on forward entropy flow and where the ultimate thermodynamic constraints on life's complexity might lie. Research into quantum coherent photosynthetic complexes already hints that coherence phenomena can transiently suppress entropy production, much like pushing a boulder uphill for a fleeting moment through quantum tunneling before gravity reasserts its inevitable downhill